Okay, let's just dive right in. Welcome to today's Explainer, where we are going to break down a truly groundbreaking leap in how AI is being used to discover new drugs. We're looking at a revolutionary framework called Mol-JEPA. Now, that stands for Multimodal Joint Embedding Predictive Architecture. And look, this isn't just some minor incremental update. We are talking about a fundamental shift in how artificial intelligence actually interprets the very building blocks of medicine.
But before we get into the nuts and bolts of the solution, I've got to ask you something. Just think about this for a second. Why do 90% of clinical drugs fail?
The harsh reality is that developing novel drugs is notoriously difficult, not to mention incredibly costly. And most of the time, drug candidates don't fail because the initial chemistry was wrong. They fail because of real-world biological hurdles. We're talking about things like weak target binding, poor absorption in the body, rapid metabolic clearance, or unexpected toxicity. Traditional computational chemistry often just hits a massive wall when it tries to predict these complex biological failures. Honestly, it's a lot like trying to predict how a sports car will handle a flooded highway by only looking at the blueprint of its engine. It's just not the whole picture.
So here is our roadmap for today. First, the drug discovery problem. Second, why current AI struggles. Third, the introduction of Mol-JEPA. Fourth, its multimodal architecture. Fifth, real-world performance results. And finally, the future of molecular AI.
Starting with number 1, the drug discovery problem, and specifically the flaws in standard AI training.
This comparison right here perfectly illustrates where standard AI goes wrong. In traditional AI, think image recognition, models use a technique called data augmentation. You can take a picture of a dog, crop it, flip it around, alter a few pixels, and the AI still learns that, yep, it's looking at a dog. But you absolutely cannot do that with molecules. If you change a single atom, or even just tweak a bond in a molecular structure, you don't end up with a slightly different version of the same molecule. You drastically alter its properties. It creates what chemists refer to as a property cliff. The problem is, traditional self-supervised learning models try to mask or alter parts of a molecular graph to teach the AI, but they end up feeding it chemically invalid, impossible structures. It's built on a completely flawed foundation.
Which brings us to part 2. Why current AI struggles so much when it views molecules in isolation. Current AI is struggling because it's essentially looking at molecules in a vacuum. Think of it like casting an actor for a movie. You really can't judge an actor's performance just by looking at a static headshot against a blank white wall, right? You need to see them in the actual environment, interacting with the set, responding to the lighting, bouncing off the other actors. Drugs are exactly the same way. Staring at a molecular structure alone just isn't enough. As this quote brilliantly points out, a drug's behavior emerges from its complex interactions with biological systems, protein networks, cellular states, whole physiological environments. If your AI only understands the structure, it is flying completely blind to the biology.
All right, part 3. Enter Mol-JEPA. This is our multimodal world model.
Let's really dig into the architecture here. Notice how, starting at the bottom, the model is taking in a vast array of different molecular modalities. We aren't just giving it a basic 2D drawing of a molecule. It's looking at atomic structures, cellular phenotypes, and complex quantum chemistry data. All of this gets processed by encoders and fed into a transformer predictor. But the real magic, the absolute game changer, happens right in that middle block, the one labeled modality masking.
Modality masking is the crucial point to understand. Remember a minute ago when we talked about how changing a molecule's atoms creates fake, invalid chemistry? Well, Mol-JEPA doesn't do that. Instead of altering the molecular graph itself, the model hides entire types of data, entire modalities, and forces the AI to predict that missing information based entirely on the remaining biochemical context. It learns by predicting massive missing pieces of a multi-dimensional puzzle. Basically, if the AI can predict how a molecule is going to behave inside a cell just by looking at its quantum properties, it has achieved a true, deep chemical understanding.
Moving to part 4, the multimodal architecture itself, which utilizes 14 different modalities.
Look at the sheer variety we're dealing with here. Mol-JEPA absorbs 14 different modalities in total. It's incredible. UMA gives it atomistic 3D foundations. Clone provides cell painting representations, which literally show the physical changes happening inside cells. GNN brings in the graph structure. XTB delivers complex quantum chemistry simulations. And Chembl feeds it real, hardcore experimental bioactivity measurements. By weaving all of these together, Mol-JEPA gets to see the molecule from every conceivable angle.
Now, to train a beast like this, the researchers had to assemble a combined data set of roughly 4.69 million unique compounds, and crucially, they incorporated those highly sought-after ADMET profiles. That's absorption, distribution, metabolism, excretion, and toxicity. Also, to make sure the AI actually learns properly, instead of, finding lazy computational shortcuts, they use something called SIGREG. That stands for Sketched Isotropic Gaussian Regularization. It sounds complicated, but you can just think of SIGREG as the strict guardrails. It keeps the AI from collapsing its learning, making sure that every single modality pulls its weight and contributes to the final understanding.
Part 5. Real-world performance results. Let's see how it actually holds up against the benchmarks.
The proof, as they say, is in the data. Looking first at the mean absolute error, or MAE, where lower is obviously better, those striped orange bars represent Mol-JEPA. You can clearly see it consistently outperforming standard models across really complex benchmark families like Expansion RX and Biogen ADME. And then, tracking out-of-distribution generalization on the right, as molecules become more and more distinct from the training data, moving left to right on that x-axis, Mol-JEPA's error rate stays remarkably low compared to the baseline models. It absolutely excels at finding truly novel drugs in uncharted chemical territory.
Breaking it down statistically using Wilcoxon sign rank tests, Mol-JEPA, sitting there on the far left, wins roughly 38% of the time. It is clearly leading the pack. But the detail the researchers really highlight is that Mol-JEPA shines the brightest in low data settings. In the incredibly expensive, high-stakes world of drug research, you rarely have millions of data points for a specific novel disease target. Having an AI that can accurately predict biological behavior with limited data, that is an absolute game changer.
So, out of all those 14 modalities, which one is actually doing the heavy lifting? Well, in a leave one modality out test where they remove just one piece of the puzzle at a time, a clear winner emerges. The big takeaway here is that the molecular graph modality at the very top, when combined with expert-designed features like MOE right beneath it, drives the absolute best results. The physical structure of the molecule is still the bedrock, for sure, but the magic really happens when the AI is forced to predict the biological context wrapped around that structure.
Finally, Part 6, the future of molecular AI, and why we need to rethink AI abilities altogether. To sum this all up, here are your key takeaways. Mol-JEPA proves once and for all that we no longer need to rely on chemically invalid augmentations to train these systems. By masking modalities instead of tweaking molecules, it consistently outperforms established baselines like ChemProp and Chameleon. But most importantly, it proves the massive, undeniable value of multimodal biochemical context. Treating a molecule like an actor in a fully realized multidimensional biological environment is the real key to unlocking true predictive power in AI.
I want to leave you with a final thought to really ponder today. If Mol-JEPA can synthesize quantum data, cell biology, and graphical structures to predict novel medicines, what other data-scarce fields could be totally revolutionized by this predictive multimodal approach? Could we use this exact same framework to discover new sustainable battery materials, or maybe novel biodegradable plastics? The architecture is there now, and honestly, the possibilities are endless.
If you learned something new today, don't forget to like, subscribe, and follow Open Life Science, OLSA, for more simple and insightful science explainers. See you in the next one.